I'm Chandre. I'm coordinator for SADC. I am now handing over to our chair and moderator, Dr. Gerald. Dr. Gerald, you are welcome.
Hello, everyone. Let me confirm you can hear me.
Yes, I can hear you.
All right, perfect. Thank you to our third webinar in data science series, and thank you for making up time to join us today. We thank the joint organization of this webinar. Our sincere gratitude to our speaker today, Dr. Julius Mulindwa from Makerere University College of Natural Sciences. The brief background to this webinar series, it is jointly developed by the Sickle Cell Disease Data Coordinating Center, SADC, and which is housed at the University of Cape Town in South Africa. And it is funded generously by the National Heart, Lung, and Blood Institute of the US NIH. It is also jointly developed by the Human Health and Heredity in Africa Consortium, that is H3 Africa, and also the African Society in Human Genetics, and as well as the Wellcome Connecting Science. The aim of this webinar is to highlight the ongoing research in data science and biomedical research, and to expose attendees to cutting-edge methods and approaches in data science and in data science for biomedical and epidemiological research in Africa. The series that started in October 2024 will run until January 2025. This webinar series is leading up to the 15th African Society of Human Genetics, AFSG, and the first Ugandan Society of Human Genetics and Bioinformatics. And I'll put the link in the chat shortly. Please feel free to apply for your travel fellowship and submit your abstract. But I know this data in past, I think, but I'll put the details to the link to the upcoming 15th Society Conference. So a brief introduction to about Dr. Mulindwa. He's a senior lecturer at the University of the Makerere University at the Department of Biochemistry, the College of Natural Sciences, where he holds a bachelor's degree in biochemistry from Makerere University. He also has a master's of science in cell biology from Helden University in Germany. And he also has a PhD in molecular biology from still the same university. His research interests are in understanding host and parasite interactions through studies on human population genetics, transcriptomics, and microbiome. Julius has undertaken such studies through the H3 Africa under the Trypanogen project, whose focus is on understanding the genetic susceptibility to neglected tropical diseases, including trypanosomiasis and schistosomiasis. He has further interest in parasite biology, where he is involved in studies on single-cell transcriptomics of trypanosomes in order to understand cell-to-cell heterogeneity. All right, that's a brief about our speaker today. Throughout the webinar, we encourage you to utilize the chat box to ask questions and also comment and make any comments along the way. At the end of the webinar, I'll put a evaluation form that you can try to give us feedback regarding today's webinar. Some housekeeping rules: try to keep yourself muted at all times so that we do not interfere with our speaker. Once again, I welcome you all, and I wish to introduce Dr. Julius Mulindwa. Over to you. Thank you very much.
Thank you very much, Gerald, for that really humble introduction. Yes, my name is Julius Mulindwa, and I would also like to thank the African Society of Human Genetics for giving me this opportunity to present our research. As Gerald mentioned, this is part of a group called a consortium called Trypanogen. For the interest of my not so good internet, I will mute my video so that it doesn't interrupt my bandwidth. I hope it's okay for everybody.
Yeah, that's fine.
So, talk about today is the genomics of a neglected tropical disease, which is schistosomiasis. Okay, so what is schistosomiasis? So this is a disease which is also known as bilharzia, and is a disease which is caused by nematode flukes, trematode flukes, and these flukes are of the genus Schistosoma. In Africa, we have three main species which affect our continent, and that is Schistosoma mansoni, Schistosoma hematobium, and Schistosoma japonicum, as you see the distribution here on the map of our continent, Africa. But this disease actually happens worldwide, but here my focus is on Africa. So it affects over 221 million people, and it mainly affects the poor communities. That's why it's categorized as one of those neglected tropical diseases. The highest prevalence of this disease is often seen in school-age children and also young adolescents. So we are looking at the age between 5 to about 19 years of age. This is where you see the highest prevalence of this disease. When it comes to its infection cycle, it starts off by individuals or people who go to water sources. These are mainly freshwater sources, which have these infective sacchari. So these sacchari are able to penetrate the skin, get into the blood system, and once they're in the blood system, these parasites mature into adults. And as adults, you have a male adult, and you do have the female adult, and these two mate, they actually do mate, and from that mating, you have eggs which are given off from the female schistosome. So once these females are formed, you do have them moving into the urinogenital tract, and this is from the schistosoma, mainly the Schistosoma hematobium, which result in this. And in the Schistosoma mansoni, you do have the eggs that are produced from that parasite moving into the intestinal tract, and that's why you do have two main forms of this disease caused by these parasites, and that is the urinogenital schistosomiasis and also the intestinal schistosomiasis. So once these eggs are in the urinogenital tract, you often release these eggs through urine. And once they're in the intestinal tract, they're often released from stool. And once individuals who are infected with these parasites encounter certain water sources, mainly freshwater sources, and they either defecate or urinate in these water sources, they release these eggs. And these eggs, once in that water source, hatch into these free-living miracidia, and these miracidia are able to infect snails. So there are different species of these snails. Is the Bronfomolia snails and the Brunel snails, which take up this miracidia and are able to release the sacchari, and the sacchari maintain this particular cycle when it comes to the intermediate host, the snails, and also the human host. So the interesting thing is that the cycle is often maintained through individuals who are able to maintain this cycle by urinating or defecating into freshwater sources. And that's where you have people having activities like swimming. Mainly children go out to this water to swim. You have mothers collecting water for cooking, for washing. You have fishermen. And all this, once they get into contact with this freshwater source, which have this sacchari, then they get infected with these parasites which cause schistosomiasis. So diagnosis of this disease mainly involves two major methods which are used for screening, especially field screening of this disease. And we have the Cato-Katz method, which involves visualization of eggs by microscopy, the eggs that you find in the fecal material. And there's also the point of care circulating cathodic antigen. These are proteins. These are antigens which are released by the parasites. And this can be detected in urine. And there's what they call a POC-CCA strip test, which is an antigen lateral flow test that is used for diagnosis of this disease. When it comes to its management, there is treatment with a drug called praziquantel, and this drug is often given annually through mass drug treatments, mass drug administration to mainly children, and it's often donated by the World Health Organization to many of these poor rural individuals through their control programs. And another method that is often used is controlling snails. And sanitation, very important, because you need to inform people that, hey, instead of going into the open water and defecating and urinating in the open water, please have a latrine where you can deposit and prevent this whole cycle and spread of this disease. And this is often through community engaging practices. So over time, we've observed, and through many surveys, we've observed that people often under the same exposure often have varying worm burden and egg shading. People who have these eggs who are having these parasites often have different eggs that are shed. And you often have categories of people who have high worm burden, and you have categories of people who have low worm burden. And we often call this high or low infection intensity with these parasites. Here we are having individuals in a population, but you have those with a high parasite intensity and those with low parasite intensity. And the question that we asked is that, how does this infection intensity affect the host? Or how does the host have factors which are able to sustain either a high infection intensity or able to exclude many of these worms? And in order to do this, we came up with a hypothesis that, hey, when it comes to the host and how the infection intensity affects the host, is it dependent on the host genotype? Are there genetic factors that make one susceptible to high or low infection intensity? Is it dependent on the microbiome? Because these eggs from this parasite, which are released through the gut, and what if this affects the gut microbiome, or the gut microbiome influencing the infection intensity? How does it influence the transcriptome of the host? And how is the immune response in presence of this different infection intensity? So the main aim of this presentation is to show you how we use the different omics approaches, microbiome, genotyping, immune response, and transcriptomes to understand how the host interacts with this parasite. So our study design was, we targeted school children, and we went to a place which is highly endemic, or actually it's a hotspot for Schistosoma mansoni, and we screened the population. Obviously, we didn't know how the population infection intensity was, because from this population of school children, we could identify children with high infection intensity, low infection intensity, and those who are uninfected in this whole population. And how we did that is first screening of these children using two methods. That is the POC-CCA, where we took a urine sample and subjected it to the POC-CCA test. So this test, essentially, these are strips. It's a lateral flow method whereby you can have an individual who is negative, uninfected, and it will show you no band at this T site. This is the test. This is the control line. Then those individuals who have high intensity and those who have trace. And this will categorize them as the low individuals with low infection intensity. So this is the first indicator that shows you of individuals who have high infection intensity, low, and those who are controls. And this is through what we call a POC-CCA urine test, and you use urine for this purpose. For the cattle-cats method, you take stool, you take the very children, tell them that, hey, go get me a sample of stool. You give them a sample tube, sample container. The stool is placed in there, and you take this stool, homogenize it, sieve, pass it through a sieve, and then get samples and align them on a slide. And on the slide, you put a cellophane tape, and you're able to observe the stool and count the eggs. You call the eggs are often excreted through the stool. So you count the number of eggs on 2 slides, and you take the mean from these 2 slides and eventually calculate the eggs per gram that you would have obtained. In addition, we took other anthropometric measurements, including the age of the children, the sex, the height, weight, and the MUAC. This is the medium upper arm circumference. These are measurements that we took that were used in the analysis. So after screening, we recruited children, and the ones who were recruited, we did some sample collection where we took stool sample, we took a whole blood sample, and we also took PAX gene blood. From there, from the stool, we extracted the fecal DNA. From the whole blood, we're able to obtain plasma and also DNA. And from the PAX gene blood, we're able to extract RNA. So what were these used for? We used the fecal DNA to carry out some microbiome 16S RNA sequencing. For the plasma, we're able to attain the infection intensity by carrying out another test. And this test is called the circulating anodic antigen. This is also an antigen that you find in the plasma from the parasite. And here you're able to quantify the infection intensity. And this is one of the tests we carried out using the plasma. Then from the whole DNA, we're able to extract the host DNA, which was used for genotyping. And for this, we did a candidate gene study with this. We'll be looking at that. And from the PAX gene blood, we're able to get the RNA, which RNA was used for RNA sequencing to attain the transcriptome for the transcriptome analysis. So first off, from the survey, what was the prevalence and what were the intensities of the infections that we found from this population? So the sample test that we carried out, like I mentioned, the first screen was by POC-CCA. We recruited children for the study, and we recruited 727. And from these 727, we were able to obtain stool samples, 554, and about 600 samples for which we did CAA analysis to obtain the infection intensity. Our sampling was done on 7 sites, and you realize that there are these 5 sites which were quite close to the lake. Actually, this is the site where we carried this from, is a district called Pakwach district. It's found in northwestern Uganda. And when you look at this map here, you're having here, this is our Lake Victoria. It's a huge lake in Uganda. You have Lake Choga here in the middle, and we have here what we call Lake Albert. From Lake Victoria, you have a river called River Nile that goes through Lake Albert and goes all the way to the Mediterranean Sea, through Sudan, through Egypt, all the way to the Mediterranean Sea. So this district here, Pakwach, has the Albert. It's at the tip of Lake Albert, and it has the River Nile that actually continues through. So we actually call this the Albert Nile. Albert, Lake Albert, and the River Nile. And we had these sites which were quite close to the Albert Nile, and we have these sites which are a bit distant from there. And the prevalence kind of correlated with the distance from the Albert Nile. We had a high prevalence in these 5 sites which were close to the Albert Nile, and we had a kind of low prevalence of the Schistosomiasis in those sites which were kind of distant from the Albert Nile. And we realized later on that we obtained most of our control samples from these sites which are a bit distant from the lake. So another thing that we observed was when we took the anthropometric measurements, we were able to obtain the height, we're able to obtain the age, and we're able to estimate what they call the height for age Z-score. And this height for age Z-score, once you have it less than 2 standard deviations, individuals who have a HAS score which is less than 2 standard deviations are often categorized as stunted. And we observed that the individuals who were cases, mainly here, the infected cases, had their mean HAS scores less than 2 standard deviations, and we kind of reported that they were stunted based on the observations that we saw. So after carrying out this, the CAA and the cattle cards test, we were able to also categorize individuals based on their infection intensity. We have individuals who had a high infection intensity, and this made up over 70%. These were 418 out of the 600. And these were categorized based on having a CAA value of over 1,000 picograms per mil and cattle cats values over 100 eggs per gram. Then we had individuals who had a low infection intensity, and these were categorized again based on CAA values and KK. And we also had about 16% of our population that we tested being controls. These were the individuals who were uninfected at all. So in summary, we found a high prevalence of Schistosoma masoni in this place, the sites that we went to. We were able to categorize the high and low infection intensities because ultimately we want to see how do these infection intensity influence the host. And we're able to observe some stunting among the children that we recruited into this study. And more details about the study can be found in this paper that we published. It's titled High Prevalence of Schistosoma masoni Infection and Stunting Among School-Age Children in Communities along the Albert Nile, Northern Uganda. And this was a cross-sectional study. So this marked the baseline where we were able to collect samples, which we proceeded with for the next parts of the study. So the next thing is that we wanted to find out how does infection intensity affect the host transcriptome. And for this, we're able to collect over 80 samples, packaged samples. However, following quality control and screening of the samples, we're able to get about 64 samples which were sequenced. Out of the 64, we had 34 of them which were high infection intensity samples. Low infection intensity were 10, and the controls were 20. So for this RNA that we extracted, libraries were made and sequencing was done on the Illumina NovaSeq, for which we're able to obtain paired end reads, 150 base pair paired end reads. And we carried out the transcriptome analysis pipeline, which essentially starts from aligning using the HiStack program to the reference genome that we use, the GRCH38. And thereafter, you obtain reads that map onto this genome. And based on the reads that you're able to obtain, you're able to carry out a differential gene expression analysis. And for this, we use the DESeq tool, which is embedded in R. So what did we observe after carrying out the differential gene expression analysis? I'm going to refer to these as DEGs. When we compared the high infection intensity versus the low infection intensity samples, we observed about 56 genes which were differentially expressed, 56 genes. 43 of them were upregulated and 13 genes were downregulated. And this is depicted here in this volcano plot. All those signals which are in black, these were not good signals. We have a log fold change, the ones which are in blue, log 2-fold change, you have a 2-fold upregulation. Then those that were significant in green here, and the ones in red are the ones that had significant p-values, and they had over 2-fold log-fold change. So we essentially looked at those that were quite significant with a high fold change, and we're able to obtain 56 differentially expressed genes. Then when we compared the low infection intensity with the control individuals, we observed that there were actually no differentially expressed genes which were quite significant. And what did that tell us? When we compare to other studies, there's a study that was carried out in Tanzania on children and adults. And in this study, they were able to identify 383 differentially expressed genes. They had 11 cases, 22 controls. And when we compare these, the DEGs that we obtained to that in a study which is part of our consortium, and in the study, they were looking at TB rhodesiense. It's another parasite altogether. It's a hemoparasite, quite acute. You also get high parasitemias. And when they did the study in Trypanosoma rhodesiense from Malawi, they used 23 cases, 21 controls, and here they saw over 3 1,000 differentially expressed genes. So what did this tell us? One is that we had very low differentially expressed genes, meaning that the parasite does not really highly affect the gene expression profile of these individuals, but this kind of expression profile is often observed in chronic infections. S. mansoni and S. hematobium are quite chronic, and these low differentially expressed genes that were observed in the blood as a result of these infections, which are quite chronic. However, the genes that we observed here were highly associated with a condition called fibrosis. How do they influence this condition called fibrosis? Is that we had genes like MALAT1. I can show it to you back here. We have MALAT1 here. MALAT1 is a long non-coding RNA, and this long non-coding RNA influences the p53 pathway, and this p53 pathway is directly involved in liver fibrosis. We also identified genes like SUZ12, SUZ12, and this one is directly involved in the SMAD and TGF pathway, and this pathway is also involved in liver fibrosis. So we found a number of genes, not so many of them, and these genes are associated with the condition of fibrosis, which is observed in schistosomiasis. So more of this work I've just presented to you. A little of it can be found in the paper published by Joyce Namulondo. She was one of our PhD students. She graduated recently, and her title was Transcriptome Analysis of Peripheral Blood of Schistosoma mansoni Infected Children from the Albert Nile region in Uganda Reveals Genes Implicated in Fibrosis Pathology. So more of this work can be obtained from this publication by Joyce. So the next thing we wanted to see is that how does the infection intensity affect the host genotype? Are there individuals who have certain genetic markers that make them susceptible to either high infection, or how can you distinguish these individuals who have high infection compared to those who are controls based on their genetic markers? So in order to do this, we obtained 296 DNA samples for which we categorize them into cases based on the top 25% based on the CAA test, and the bottom 25%, 148 samples of these, and we categorize them as the control samples. So for this, we did genotyping, and the design that we used was a case control study. We had cases, we had controls, and we genotyped the samples on the Illumina H3 Africa chip. And for this, we went for a candidate genes, and we had 24 candidate genes. Many of these were informed based on literature and information that was known about these genes as being involved in susceptibility to Schistosoma mansoni. So we carried out SNP calls using Genome Studio. We did haplotype calls, and we also did equital analysis using the Plink. So what did we get? We obtained several significant SNPs, and we also obtained several significant haplotypes, and all these SNPs and haplotypes are associated with infection. They are associated with infection. And the interesting thing that we see here is that we have IL-6 here, which happen to have a high frequency and has SNPs which are highly significant as associated with infection. And when we go into that locus, the IL-6 locus, you're able to observe how the SNPs are in the locus. And how the SNPs are, you have the significant EQTLs and also the significant SNPs. And these are depicted by the Manhattan here, these peaks that you're able to see, the signals that you're able to see. And they are also depicted in this region based on the DNase count and DNA signal. DNA signal shows that this is euchromatin, which is readily expressed as compared to heterochromatin, which is often closed up. If it's a closed up region, you will have spaces like this because it's highly closed up. But then if it's euchromatin that is highly expressed, then you have this gene which is highly expressed. And based on this, it was suggestive that the SNPs in IL-6 locus were functional and they could play an important role in the response. Being a cytokine, IL-6 being a cytokine, it plays an important immune response to schistosomiasis infection. So a lot of this work was published by Oscar Nyangiri, and the paper is titled Variants in IL-6, IL-10, FCN2, RNase 3, IL-12B, and IL-17 loci are associated with schistosoma masoni 1 burden in the Albert Nile region of Uganda. So having identified the genotypes which are associated with infection, we then looked at the immune response. How does the host respond to the presence of this parasite in the blood? In order to do this, we obtained the plasma, about 142 samples, and these were categorized into the high infection intensity, 56 samples, low infection intensity, 41, and the controls, which were 45. So for this, we did the systemic cytokine level analysis, and we used the Luminex assay, and it was a 13-plex assay where we looked at the Th1 and Th2 cytokines. The Th1 cytokines included interferon gamma, interleukin-2, IL-10, IL-12, P70, TNF alpha, and TNF beta. And for the Th2, we looked at the GMSCF, IL-1B, IL-4, IL-5, IL-6, IL-9, and IL-13. So these cytokines are involved in response to the schistosomiasis. Luminex MAGPICs in order to obtain the mean fluorescence intensities from the samples that we obtained. So what did we observe? If you look at the cytokines, starting off from interferon gamma, we saw no differences in the cytokine levels with the high, low, and the controls. But then when we looked at several of the other cytokines, be it Th1 or Th2, we found a really significant difference between the high infection intensity samples as compared to the low and the control samples. So high is here in red, the blue are the low infection intensity samples, and the green are the control samples. We really saw a huge, significant difference between those samples. When you look at IL-10, wasn't a significant difference for this particular cytokine. GMSCF, interleukin-1, IL-9, IL-6, IL-5, IL-4, and IL-13, which saw a really huge, significant difference between
Hello, we are sorry, we have lost connection to Dr. Julius, but we are trying to get in touch and see if he can rejoin the call. Sorry about that.
My apologies there. My network seems to be on and off.
Sorry about that.
Yeah, so let me reshare my slides. My apologies. I'm just inquiring where I had stopped, where the network went off. Hello? Hello, can you hear me?
Yes, we can hear you. In fact, you went off. Can you go on the current slide? I'll check by the slide where you are.
I was looking at the immune response here.
Yeah,
Is
you
this
are
where-
summarizing. Yeah, you are summarizing this slide
Okay,
here.
okay. Thank you. Apologies for this. Okay, so the summary from this is that there was immunosuppression in most of these children who had a high infection intensity because it was quite evident based on what we observed. So we looked at, just to confirm that we're saying that we're highly low cytokine levels in the low and control individuals, we looked at their expression data. And this expression data was from Joyce's data, where she looked at the transcriptome. And when we looked at the genes and we looked at a PCA plot of these genes, of only the cytokine genes, cytokine and cytokine receptor genes, about 176 of them, you observe here that in red, we have the high infection intensity. In blue, we have the low, and the green are the control samples. Many of the samples here are below PC1 and are below 0 when it comes to PC1 and PC2, and it was indicative of low gene expression of these particular cytokines in these individuals with high infection intensity, as compared to the green here, which you have it as high. And the same information can be observed when we plot the normalized read counts of each of these cytokines. And we observe that they are much lower in the high infection intensity groups as compared to the low and the control individuals. So immunosuppression in these children with a high infection intensity was evident, and we were able to observe it both from the cytokines in the plasma and also looking at the gene expression information. So the next thing that we wanted to look at is how does the infection intensity influence the gut microbiome? And in order to do this, we extracted about 140 samples, fecal DNA from 140 samples. Of these, we had high infection intensity, 58 of them. Low infection intensity were 37, and the control group had 45 individuals. So we did 16S ribosomal RNA sequencing of the V3, V4 region. And for this, we use the DNA nanoball. This is a technology which is found at the Big EI. And from this, we're able to obtain 300 base pair paired end reads. We did OTU and taxa annotation using the DADA2 tool. We did diversity analysis. This is alpha and beta diversity using the phyloseq tool. And then we also did differential abundance analysis using LF, SE, DE-seq2, and also HR. Then eventually we also did some linear regression association analysis as we'll see with the results. So what did we obtain? We observed differences here at the phyla and genus level. We're able to observe differences between the high infection intensity, low, and also the control samples. And this is evident by the profiles that we observe for the specific genus within each of these groups. And we'll be looking at that in the next slide. This was evident of some microbiome dysbiosis between the individuals. So when we did differential genus enrichment in the gut microbiome of these individuals, we're able to see significant enrichment. For the genus Prevotella, we saw enrichment. We saw enrichment in comparison to the control groups in Fasciculobacterium, in Alloprovotella, and also in Ruminobacter. And this enrichment was also highly correlated with infection intensity. We saw a high correlation and association of this bacteria genus with the infection intensity. So these bacteria happen to be gram-negative anaerobes that generate short-chain fatty acids. So enrichment of these short-chain fatty acids is often associated with an anti-inflammatory activity by inhibiting the NF-kappa B. The NF-kappa B pathway is involved in generation of immune cytokines. So having high short-chain fatty acids results in an anti-inflammatory effect, and this could be one of the plausible explanations as to why we're having low levels of cytokine response when it came to the individuals with a high infection intensity. So again, comparing the gut microbiome with the immune response, we did an association analysis between the abundant genus and the cytokines that we observed that we were able to measure using the Luminex assay. And for this, we did a fixed linear regression analysis, whereby we obtained one genus, compared it with one cytokine, and we had the other 12 cytokines as covariate. Significant associations have less than 0.05. And what do we see? We saw the interesting cytokine IL-6, which was identified by Oscar from the genotyping analysis. And we observed that it was highly associated with this genera, Alloprovotella, Gastroenophiles, and the Streptococcus. So we found some association between the immune response, the IL-6 that was also identified as a key locus, key gene which has significant locus of association, also having it associated with specific bacteria genus, which are also involved in which are also enriched in individuals with a high infection intensity. So this work involving the gut microbiome and also immune response was mainly carried out by Ibra Rujumba and also Caroline Musime, who are based at the Center of Excellence at Makerere University. And this work has been submitted to the BMC Microbiology Journal, and it's titled High Schistoma Mansoni Infection Intensity is Associated with Distinct Gut Microbiome and Levels of Systemic Cytokines in Children along the Albert Nile in Uganda. So in conclusion, we started off by trying to understand how is the Schistosoma mansoni infection intensity affected by the host microbiome, or how does it influence the host transcriptome, how does it influence the immune response, and how does it influence the genotype of the individual who is infected. And in order to have a conclusion to this, we need to first understand the pathophysiology of these parasites. Once this parasite gets into the blood system, we often have the male and the female parasite mating, generating eggs, which eggs are then transported across into through the mesenteric vessels, getting to the intestinal lumen, and in the intestinal lumen, you often have bacteria there. So we looked at the microbiome, the different bacteria that you have within the intestine, and how these different bacteria in the microbiome are influenced by the infection intensity. So once these eggs have crossed, they often form granulomas, and presence of these granulomas results in an hyperimmune response. And how this hyperimmune response is counteracted by the host is through the expression of these B and T cell regulatory factors. These Tregs and Bregs are involved in suppression of the hyper response, and that is probably one of the reasons why we found a low immune response from individuals who had a high infection intensity. So from the transcriptome, we observed that the differentially expressed genes were highly associated with fibrosis. There were few, but these were genes which are highly associated with fibrosis. When it comes to the immune response, there was hyperimmunosuppression in the high intensity individuals. And when it came to the microbiome, we've observed that there was microbial dysbiosis, and there was specific enrichment of particular genus in individuals either with a high, low as compared to the control individuals who had no Schistosomiasis infection. And when it comes to genetic susceptibility, we have identified genotypes and haplotypes and also expression quantitative loci, which are associated with Schistosomiasis 1 burden. So in conclusion, looking at the genomics of schistosomiasis, we have identified genetic markers, we have identified biomarkers, which are crucial probably in the control of this neglected tropical disease. So I would like to acknowledge these groups from Makerere University, headed by Professor Enoch. We had Oscar there, we had Joyce, and a number of people from his group. From the University of Glasgow, we have Professor Annette McLeod. From the University of Liverpool, we have Dr. Harry Noyes, who is involved centrally in mentoring individuals for bioinformatics. At the MRC LSTMH UVRI, we had Professor Alison Elliott and Jacent, who are mainly involved in the CAA analysis. The vector control team, headed by Mr. Adrico Moses, were involved in the field work, KK analysis, and the screening. The district health officials at the Pakwach district, IDI, Makerere University, through the Center of Excellence, headed by Dr. Daudi, where you have Ibra and Caroline, who are involved in the microbiome analysis, and also the Ministry of Health, IDRC, CPHL, headed by Dr. Isaac, who are mainly involved in the Luminex assay and analysis. Finally, I would like to acknowledge our funders, that is the Wellcome Trust through the Science for Africa Foundation, who funded the Trypanogen project, and also the Makerere Research Innovation Fund, which provided funds for the analysis, mainly on the microbiome work that we did. We also acknowledge the participants. Here were under a tree, sensitizing our participants, mainly the school-age children who were involved in the study. And this is a glimpse of where they actually go. You see activity here. You see these children who are in the water there. You see these are headed out to go fishing. And you see the mothers here coming to collect water that they use for the cooking. So thank you very much.
Thank you very much, Julius, for the wonderful presentation. I would like to appreciate that from your side and also from the developers and organizers of the webinar. Just my personal appreciation, thanks for building the omics capacity, especially using a tropical neglected disease such as schistosomiasis. Thank you very much, and give it up. I know there are a couple of questions. I don't know if it is okay for you to pick those that are very pertinent and you try to respond to them, since we have 8 minutes to go. Is that okay?
Yes, please. You can.
Okay.
I'll look up for them.
Okay.
So I have a question here. How did you control for possible differences in the parasite genotypes? I'd like to mention here that we looked at the host, the human genotypes, and we did not look at the parasite. We didn't do any analysis on the parasite. We have a question here. Correlation between intensities of infection measured by, okay, that was from Harry. I'm trying to look out for a question here. I would like to know if you will share the presentation. It was recorded and the recording will be shared by the organizers. Yeah, trying to look out for questions here.
Can I ask mine as you look out for?
Yeah, please do.
Okay, so it is more the translational impact part. What are the current gaps in translating genomic data on schistosoma species into actionable interventions such as diagnostics, vaccine, and treatment strategies?
So some of the challenges involve validation. If you noticed, we do have small sample sizes that we carry out these analyses on, and these analyses are kind of like discoveries. So when you discover something, you often need to validate it using a larger sample size, and this larger sample size often comes at a cost, which cost is not often met. So translating this into diagnostics, translating it into other forms of intervention, let's say vaccines and all that, it requires more work, it requires validation, it requires an expense. And that is something that we are trying to venture into. And as researchers, we first start off with the discoveries, then we can move on to the next steps. But it's somewhat a challenge.
All right, and then one more question. I'm being very greedy. So it's regarding collaboration and capacity building. In your view, how can African researchers and institutions collaborate effectively to build capacity and ensure equitable access to genomic technologies for schistosomiasis research?
So the H3 Africa was a really good consortium where a lot of us met. I joined the H3 Africa through Tripanogen. We had 2 phases, Tripanogen 1 from 2014, and then we had another phase that began in 2018, ended in 2023. But within there, we built remarkable networks. And through even the H3 Bionet, headed by Nikki, we were able to get resources, analysis resources, pipelines. Some of these were not in place, but through collaborations, centers of excellence, including one here locally at Makerere University, headed by Daudi, you're able to have resources and techniques which are passed on even to other scientists. So my answer to that is through collaborations. I might have a certain resource, but a neighbor, let's say in Sudan, might not have it. So through collaboration, we're able to network, and we're able to build capacity where it's missing.
Okay, so let me take one more question. I think we have 4 minutes to go. I think there's a question here in the chat, which says, were there some antigens in the plasma collected associated with the expression profiles or severity of the infection?
Were there antigens in the plasma?
In the plasma
that were associated. So when we got the plasma, what we used the plasma for was for analyzing for infection intensity. And the antigen that we used is one of those antigens which is regurgitated by the worm, and that is the circulating anodic antigen. And that is the one antigen we use to look at the infection intensity. What we use the plasma for, the other thing was for the immune response cytokines, but we did not exhaust all the factors that are in the plasma. For instance, there are T regulatory cells, the Tregs, and these Tregs are often involved in suppression of the effect of the immune, of that, mainly the Th1 and Th2 responses. So that is something that we did not look out for, and those are some of the limiting factors when it comes to this research. You want to do a lot, but you're limited by resources to do one specific thing. So we carried out the infection intensity analysis using CAA, and were able to obtain the immune response by analyzing the cytokines, but there's still a lot more we can still analyze since we do have the plasma in our archives.
All right, one more regarding, it's still aligning with the same question. It is from, I think, Emmanuel Adai. It says in your conclusion, you made a mention of Tregs and Bregs implicated in high intensity individuals. Do you think checkpoint inhibitors would have an impact on lessening the infection?
Yes, there are quite a number of factors that we haven't looked at. We just looked at a few. And for instance, we have seen that we do have individuals who have a high intensity infection and they're having low levels of these Th1 and Th2 cytokines. So that is a starting point. We can again look at those very samples and then look at other factors in there. There are also factors to do with the epigenetics. Epigenetics is also associated with specific microbiomes that individuals could have. Those are factors that we didn't look at. So there's a lot of there are so many other parameters which we haven't looked at, but in such a study where you're able to identify or come across interesting findings, you can then know where else can I look. I've looked at this aspect, so where else can I look to complete my story?
All right, so thank you, Julius. And one final question still from me. What is next in your team? I know I can see Caroline, Oscar, Joy, everyone who is here, who has been part of initially Tripanogen and now your current research team, which also encompasses the team at S. So what is next? What can we expect from your team?
So like the theme is, we're going into the era of data science. We collect the data, we do not exhaust it. We collect the samples, we haven't exhausted all the parameters. So the next level is completing our hypothesis through analyzing other factors, analyzing other systems, analyzing other pathways, building networks, and see how we can use the data science resource and also our samples that we have to complete the story. So we are applying for more grants, we're applying for more funding, just to see how we can explore the research questions which we haven't fulfilled at the moment.
Thank you very much. You have reached at the top of the hour. On behalf of the organizers, I would like to express my sincere gratitude to our speaker, Dr. Julius Mulindwa from Makerere University, and to all you participants. We look forward to the next webinar. Thank you all for attending today's webinar, and please stay well. Thank you and bye-bye.
Thank you,
Bye-bye.
Dr. Julie and Dr. Gerald. We also would like to remind to complete that have been posted within the chat. And thank you once again to all participants. Thank you for today's webinar. Goodbye.